antigenic changes in the viral surface glycoproteins make influenza viruses formidable challenges for control efforts. So we have three immunologic types for influenza. We have influenza type A, type B, and type C. If it's type A, then this is the type which is antigenically highly variable and is responsible for most cases of epidemic influenza. For type B, it may also exhibit antigenic changes, but it's not the same as type A. And type B can also sometimes cause epidemics. For type C, these are antigenically stable, and it causes only mild illness in immunocompetent individuals. For the properties of orthomyvirus, as mentioned, there are three immunologic types. And influenza A strains are also known for aquatic bird, chicken, ducks, pigs, horses, and seals. Some of the strains isolated from animals are antigenically similar to strains that are circulating in the human population. So here are the important properties in this table. With its outstanding characteristics is of course the genetic reassortment. This is common among the genus Orthomyxo, and influenza viruses causes worldwide epidemics. So the rest of the properties are there in the table. For its structure and composition, influenza virus particles are usually spherical and about 100 nanometers in diameter, and although variants may display great variation in sizes. The single-stranded negative sense RNA genomes of influenza A and B viruses occur as 8 separate segments. For influenza C naman, it contains only 7 segments since it lacks the neuraminidase gene. And sizes and protein coding assignments are known for all the segments. So if we're going to talk about the segment number, then type C, influenza type C does not have the segment number 6, since the number 6 is the one with the neuraminidase. Okay. So most of the segments code for a single protein, and the first 12 to 13 nucleotides at each end of the genomic segment are conserved among all 8 RNA segments. And these sequences are important in viral transcription. Influenza virus particles contain 9 different structural proteins. So we have nucleoprotein associates with the viral RNA to form a ribonucleoprotein that is 9 nanometers in diameter and assumes a helical configuration and forms the viral nucleocapsid. There are also 3 large proteins, namely PB1, PB2, and PA. These are bound to the viral RNP and are responsible for RNA transcription and replication. Another protein would be the matrix protein, M1, which forms a shell underneath the viral lipid envelope, and it is important in particle morphogenesis and is a major component of the virion. A lipid envelope derived from the cell surrounds the virus particle, and 2 virus-encoded glycoproteins known as the hemagglutinin and neuraminidase are inserted into the envelope and are exposed as spikes. So these are surface glycoproteins, about 10 nanometers long on the surface of the particle. And this is the one that determines the antigenic variation of the influenza viruses. So this is the one that undergoes mutation, the HA and the NA, hemagglutinin and neuraminidase. And the HA represents about 25% of viral protein and the NA about 5%. The M2 ion channel protein and the NS2 protein are also present in the envelope, but at only a few copies per particle. So because of the segmented nature of the genome, when a cell is co-infected by 2 different viruses of a given type, mixtures of parental gene segments may be assembled into progeny virions. So that's why there is this genetic reassortment, because there will be changes, especially if different viruses were to infect a single individual. Influenza viruses are relatively hardy in vitro and may be stored at 0 to 4 degrees Celsius for weeks without loss of viability. Lipid solvents routine denaturants, formaldehyde, and irradiation destroy infectivity. Both infectivity and hemagglutination are more resistant to inactivation at alkaline pH than at acid pH. For the classification, the genus influenza virus A contains human and animal strains of influenza type A. For influenza virus B, it contains only the human strains of type B. And influenza virus C contains influenza type C viruses of humans and swine. Antigenic differences exhibited by two of the internal structure proteins, which is the nucleocapsid and the matrix protein, are used to divide the influenza viruses into the different immunologic types. But these proteins possesses no cross-reactivity among the three types. So the antigenic variation is based in the surface glycoproteins. So this is how we subtype the type A viruses. And the standard nomenclature for influenza virus isolates will include the following information. First would be the type, then the host of origin, if it's from a swine, an avian, or from human, geographic origin, where is the location where this was isolated, if it's in Hong Kong or in the Philippines, strain number, and the year of isolation. So where am I? Antigenic descriptions of the HA and NA are given in the parentheses for type A. So anak ng mga nababatayan yung mga H1N1, sorry, that's the parenthesis part na. Hindi describe na dito ang hemagglutinin and neuraminidase. So here's an example. The host of origin is not indicated for human isolates such as A, Hong Kong, O3, 68, and H3N2. The 68 was the year it was isolated. But it is indicated for others. So may iba na if it's from human isolates ngayon, we don't state na nga it's from human. So if it's from other sources, if it is isolated from other forms of animal, then yun, we indicate that it's from swine, the place, yun. So far, 18 subtypes of hemagglutinin and 11 subtypes of neuraminidase in many different combinations have been recovered from humans and animals. The Orthomyovirus family also contains the genus Togotovirus, members of which are not known to cause disease in humans. For the structure and function of hemagglutinin, this protein of influenza virus binds virus particles to susceptible cells and is the major antigen which neutralizing antibodies are directed. So this is the function of the hemagglutinin protein, is the attachment of the virus to the susceptible cell. Variability in hemagglutinin is primarily responsible for the continual evolution of new strains and subsequent influenza epidemics. So here's a influenza virus hemagglutinin and neuraminidase surface glycoproteins. So these are the primary structures. And as we can see here, oh, by the way, primary sequence of hemagglutinin contains 566 amino acids. And as we can see in the hemagglutinin, there is a part wherein there's supposedly proteolytic activation cleavage. And this cleavage, which cleaves the hemagglutinin into HA1 into HA2, is actually necessary for virus to be infectious. But even though it is being cleaved, it is the two subunits, HA1 and HA2, is still linked with a disulfide bond. Okay. For neuraminidase naman, there's no cleavage necessary. So here is a three-dimensional structure of the hemagglutinin protein, and it has been revealed by X-ray crystallography. So the hemagglutinin molecule is folded into a complex structure with 5 major antigenic sites, which, as you can see in the figure, it is it is shaded and is named sites A, B, C, D to E. Okay. So the HA spike on the virus particle is a trimer, and it is composed of three intertwined HA1 and HA2 dimers. The trimerization imparts greater stability to the spike than could be achieved by a monomer. And the cellular receptor binding site is a pocket located at the top of each large globule. And this pocket is inaccessible to antibody. The cleavage that separates the HA1 and HA2 is necessary for the virus particle to be infectious and is mediated by cellular proteases. Influenza viruses normally remain confined to the respiratory tract because the protease enzyme that cleave the hemagglutinin are expressed only at those sites. The amino terminal of the HA2, which is generated by the cleavage, is necessary for the viral envelope to fuse with the cell membrane, which is, of course, is an essential step in the process of viral infection. Low pH triggers a conformational change, which activates the fusion activity. For neuraminidase, the antigenicity of neuraminidase, which is the other glycoprotein on the surface of the virus, is also important in determining the subtype of influenza virus isolates. And the neuraminidase spike on the virus particle is a tetramer composed of 4 identical monomers. And there is a slender stalk, which is topped with a box-shaped head, as seen in the figure. There is a catalytic site for neuraminidase on the top of each head, so that each neuraminidase spike contains 4 active sites. And the function of this NA is at the end of the viral replication cycle. It is a sialidase enzyme that removes the sialic acid from glycoconjugates, and it facilitates release of the virus particles from infected cell surfaces during the budding process, and it prevents self-aggregation of virions by removing the sialic acid from the glycoproteins. It is also possible that NA helps the virus negotiate through the mucin layer in the respiratory tract to reach the target epithelial cells. Now, for the antigenic drift and antigenic shift, influenza viruses are remarkable because of the antigenic changes which occurs on the surface glycoproteins. And the antigenic variants of these viruses have a selective advantage over the parental virus in the presence of antibody directed against the original strain. And this phenomenon is responsible for the unique epidemiologic features of influenza. Other respiratory tract agents do not display this significant antigenic variation. The two surface antigens of influenza undergo antigenic variation, which is independent of each other. If there is only minor changes for the antigenicity of the orthomyvirus, then it is known as antigenic drift. But if it's major, then it's antigenic shift, which would result in the appearance of new subtype. And this antigenic shift is most likely to result in an epidemic. So antigenic drift is caused by the accumulation of point mutations in the gene, resulting in amino acid changes in the protein. Sequence changes can alter antigenic sites on the molecule such that a virion can escape recognition by the host immune system. So the immune system does not cause the antigenic variation, but rather functions as a selection force that allows new antigenic variants to expand. A variant must sustain 2 or more mutations before a new epidemiologically significant strain emerges. Antigenic shift naman reflects drastic changes in the sequence of viral surface protein caused by genetic reassortment between human, swine, and avian influenza viruses. As mentioned a while ago, if there are 2 different influenza viruses of the same type that enters an individual, then there can be genetic reassortment, and through the genetic reassortment, there will be changes in the production of the surface. I mean, in the antigenicity of the surface proteins. For influenza B and C viruses naman, they do not exhibit antigenic shift because few related viruses exist in animals. So oftentimes it is the type A influenza that has this genetic reassortment. For influenza virus replication, the viral multiplication cycle proceeds rapidly, and there is the shut-off of host cell protein synthesis by about 3 hours after the infection, permitting selective translation of viral mRNAs. New progeny viruses are produced within 8 to 10 hours. So what happens here is after endocytosis, after the virus is being endocytosed, the viral ribonucleoprotein complexes are released into the cytoplasm and then transported to the nucleus. So in the nucleus, the replication and transcription will take place. So mRNAs are then exported to the cytoplasm for translation. Then the early viral proteins required for the replication and transcription, which includes the nucleoprotein and the polymerase protein, are then transported back to the nucleus. So the templates, the single-stranded RNA, are copied by the RNA polymerase activity of the PB1 protein, and then some of these new genome segments will serve as template for the synthesis of more viral mRNA. So here is a more detailed explanation for the replication. For the attachment, it attaches to the cell surface sialic acid via the receptor site, which is located on the large globule of the hemagglutinin. Virus particles are then internalized within the endosomes through receptor-mediated endocytosis. So after that, there will be a fusion between the envelope of the virus and the cell membrane, which will trigger the encoating. The low pH within the endosome is required for virus-mediated membrane fusion that releases viral RNPs into the cytosol. Acid pH will cause a conformational change in the hemagglutinin structure to bring the HA2 fusion peptide in correct contact with the membrane, since at first, this fusion peptide is hidden and needs a change in the structure of the hemagglutinin in order for it to be exposed. The M2 ion channel protein present in the virion permits the entry of ions from the endosome into the virus particle, which will trigger the conformational change in the hemagglutinin. Viral nucleocapsids are then released into the cell cytoplasm. Then, after being released, then transcription and translation will then take place in the nucleus. So transcription mechanism used by Orthomyovirus differ markedly from the other RNA virus in that cellular functions are more intimately involved. Viral transcription occurs in the nucleus, and the mRNAs are produced from the viral nucleocapsids. The virus-encoded polymerase, which consists of a complex of 3 P proteins, is primarily responsible for transcription, and its action must be primed by scavenged, capped, and methylated 5' terminals from cellular transcripts that are newly synthesized by cellular RNA polymerase II. So this explains why influenza virus replication is inhibited by dactinomycin and alpha-amantine, which block cellular transcription. But other RNA viruses are not affected because they do not use cellular transcripts in viral RNA synthesis. 6 of the genome segments yield monocystronic mRNAs that are translated in the cytoplasm into 6 viral proteins. The other 3 transcripts undergo splicing, each yielding 2 mRNAs that are translated in different reading frames. At early times after infection, the NS1 and NP proteins are preferentially synthesized, and at later times, the structural proteins will be synthesized at higher rates. The 2 glycoproteins, HA and NA, are modified using the circulatory pathway, and the influenza virus non-structural protein NS1 has a post-transcriptional role in regulating viral and cellular gene expression. The NS1 protein binds to the polyase sequences, which inhibits pre-mRNA splicing and inhibits the nuclear export of spliced mRNAs, which ensures a pool of donor cellular molecules to provide the cap primers needed for viral mRNA synthesis. The NS2 protein interacts with the M1 and is involved in the nuclear export of the viral RNAs. For viral genome replication, it is accomplished by the same virus-encoded polymerase that it was involved in transcription. And the mechanism that regulate the alternative transcription and replication roles of the same proteins are related to the abundance of one or more of the viral nucleocapsid proteins. As with other negative strand viruses, templates for viral RNA synthesis remain coated with the NP. The only completely free RNAs are those that are mRNA. And the first step in genome replication is the production of the positive strand copies of each segment. These anti-genome copies differ from mRNA at both terminals. The 5' ends are not capped, and the 3' ends are neither truncated nor polyadenylated. And these copies will serve as templates for the synthesis of 3 full copies of genomic RNAs. Because there are common sequences at both ends of all viral RNA segments, they can be recognized efficiently by the RNA synthesizing machinery. Intermingling of genome segments derived from different parents in co-infected cells is presumably responsible for the reassortment, for the genetic reassortment that is typical of influenza viruses within the genus. Frequency of reassortment is as high as 40%. For maturation, the virus matures by budding from the surface of the cell. Individual viral components arrive at the budding site by different routes. Nucleocapsids are assembled in the nucleus and move out to the cell surface. And the glycoproteins HA and NA are synthesized in the endoplasmic reticulum. They are modified and assembled into trimers and tetramers respectively and are inserted into the plasma membrane. The M1 protein serves as a bridge linking the nucleocapsid to the cytoplasmic ends of the glycoproteins, and the progeny variants will bud off the cell. During these events, the HA is cleaved into HA1 and HA2 if the host cell possesses the appropriate proteolytic enzyme. The NA removes the terminal sialic acid from the cellular and viral surface glycoproteins, which facilitates the release of the virus particles from the cell and prevents their aggregation. Many of the particles are not infectious. Particles sometimes fail to encapsulate the complete set of genome segments. Frequently, one of the large RNA segments is missing. These non-infectious particles are capable of causing hemagglutination and can interfere with the replication of the intact virus. Reverse genetic systems that allow the generation of infectious influenza virus from cloned cDNAs of viral RNA segments are available and allow for mutagenesis and functional studies. Now, for influenza in humans, we have a table here comparing the viruses that infect the human respiratory tract. So it's not just the influenza virus that has an effect in the respiratory tract. There are also other viruses. So we have here RNA viruses and DNA viruses and other disease that they produce. Influenza virus spreads from person to person through droplets or through contact with contaminated hands or surfaces. A few cells of respiratory epithelium are infected if deposited virus particles avoid removal through the cough reflex and they are not being neutralized by the immunoglobulin A that is pre-existing in the epithelium, or if there is no inactivation by non-specific inhibitors in the mucous secretion. Progeny variants are soon produced and spread to adjacent cells where the replicative cycle is repeated. Okay. Viral NA lowers the viscosity of the mucous film in the respiratory tract, which lay bare to the cellular surface receptors and will promote the spread of virus-containing fluid to the lower portions of the tract. The incubation period from exposure to virus and the onset of illness is from day 1 to day 4, depending on the size of the viral dose and the immune status of the host. There's also viral shedding that starts the day preceding the start of the symptoms, and will peak within 24 hours. And it will remain elevated for 1 to 2 days and will decline after 5 days. So the infectious virus is very rarely recovered from the blood. And interferon is detectable in respiratory secretions about 1 day after viral shedding begins. So influenza viruses are sensitive to the interferon. And it is believed that the innate immunity response contribute to host recovery from infection. Specific antibody and cell-mediated responses cannot be detected for another 1 to 2 weeks. And influenza infections cause cellular destruction of the superficial mucosa of the respiratory tract, but do not affect the basal layer of the epithelium. Complete reparation of cellular damage probably takes up to 1 month, and edema and mononuclear infiltrations in response to cytokine release and cell death caused by viral replication probably account for local symptoms, and the fever and systemic symptoms associated with influenza reflect the action of the said cytokines. So influenza attacks mainly the upper respiratory tract. So it poses a serious risk for elderly adults, very young children, and people with comorbidities, diabetes, cancer, or immunosuppression. So for uncomplicated influenza, symptoms of classic influenza usually appear abruptly, which will include the chills, headache, dry cough, and followed closely by high fever, generalized muscular aches, malaise, and anorexia. The fever usually lasts 3 to 5 days, as do the systemic symptoms. Respiratory symptoms typically last another 3 to 4 days. The cough and weakness may persist for 2 to 4 weeks after major symptoms subside. Mild or asymptomatic infections may also occur, and these symptoms may be induced by any strain of influenza A or B. In contrast, influenza C rarely causes the influenza syndrome, and it only causes instead a common cold illness. Coryza and cough may last for several weeks. Clinical symptoms of influenza in children are similar to those in adults, although children may have higher fever and they can have gastrointestinal manifestations such as vomiting. Febrile seizures can also occur. Influenza A viruses are an important cause of croup, which may be severe in children younger than 1 year of age. And uncomplicated influenza may also cause otitis media to develop. When influenza appears in the epidemic form, clinical findings are consistent enough that the disease can be diagnosed presumptively. However, right now, we cannot presume that this is, if we have an epidemic that is that have a symptom for influenza, we cannot presume that it is caused by influenza virus, since we are currently in an epidemic caused by COVID-19. So we still need to consider COVID-19 regardless of the symptoms. If the symptoms of the patient is just like flu, we would still consider COVID-19. We would consider them as COVID-19 suspects. Sporadic cases cannot be diagnosed on clinical grounds because disease manifestation cannot be distinguished from those caused by other respiratory tract pathogens. However, those other agents rarely cause severe viral pneumonia, which can be a complication of influenza A virus infection. Pneumonia. These serious complications usually occur only in elderly adults and debilitated individuals, especially those with underlying chronic diseases. Pregnancy also appears to be a risk factor for lethal pulmonary complications, and the lethal impact of an influenza epidemic is reflected in the excess deaths caused by pneumonia and cardiopulmonary disease. Pneumonia complicating influenza infections can be viral, secondary bacterial, or a combination of the two. Increased mucus secretion helps carry agents into the lower respiratory tract, Influenza infection enhances susceptibility of patients to bacterial superinfection. So, the enhanced susceptibility of the patient to bacterial superinfection is attributed to the loss of ciliary clearance, the dysfunction of phagocytic cells, and provision of a rich bacterial growth medium by the exudates caused by the alveoli. I mean. Bacterial pathogens are most often Staphylococcus aureus, Streptococcus pneumoniae, and H. influenzae. Combined viral bacterial pneumonia is approximately 3 times more common than primary influenza pneumonia, and a molecular basis for a synergistic effect between virus and bacteria may be that some S. aureus strains secrete a protease able to cleave the influenza HA, thereby allowing production of much higher titers of infectious viruses in the lungs. Raynaud syndrome is an acute encephalopathy of children and adolescents, which is usually between 2 and 16 years of age. Mortality rate is high, and the cause of this syndrome is unknown, but it is a recognized rare complication caused by influenza B, A, and herpes virus varicella zoster infection. There is an epidemiologic relationship between the use of aspirin, salicylate, and subsequent development of the Raynaud syndrome. And the incidence of the syndrome has decreased with the reduced use of salicylates in children with flu-like symptoms. Immunity to influenza is long-lived and subtype specific. Whereas antibody against hemagglutinin and neuraminidase are important in immunity to influenza, antibodies against the other virus-encoded proteins are not protective. Resistance to initiation of infection is related to antibody against the HA, but decreased severity of disease and decreased ability to transmit virus to contacts are related to the NA. So again, if initiation of infection, we want antibody against the hemagglutinin. Since hemagglutinin again is the one that is used for the attachment of the virus to the cell surface. Okay. Protection correlates with both serum antibodies and secretory IgA antibodies in nasal secretions. The local secretory antibody is probably important in preventing infections. Serum antibodies persist for many months to years. Secretory antibodies are of shorter duration, which is usually only several months. Antibody also modifies the course of illness. A person with low titers of antibody may be infected, but will experience a mild form of disease. Immunity can be incomplete, and reinfection with the same virus can occur. The 3 types of influenza viruses are antigenically unrelated, so they cannot induce cross-protection. When a viral type undergoes antigenic drift, a person with pre-existing antibody to the original strain may have only mild infection with the new strain. Subsequent infection or immunization reinforce the antibody response to the first subtype of influenza experienced years earlier, which they call original antigenic sin. The primary role of cell-mediated immune responses in influenza is believed to be clearance of an established infection because the cytotoxic T cells will lyse the infected cells. So cytotoxic T lymphocytes response is cross-reactive and appears to be directed against both internal proteins and the surface glycoproteins. For the laboratory diagnosis, we can use the polymerase chain reaction, the rapid test based on detection of influenza RNA in clinical specimens using reverse transcription polymerase chain reaction are preferred for the diagnosis of influenza. So RT-PCR is rapid, sensitive, and specific. Multiplex molecular technologies are available, which allows for the rapid detection of multiple pathogens in a single test. And by the way, the best specimen for diagnostic testing will be the nasopharyngeal swabs or the nasal aspirates or even the lavage, the nasal lavage. And this should be obtained within 3 days after the onset of the symptom. We can also do isolation and identification of virus. The sample to be tested for virus isolation should be held at 4 degrees Celsius until inoculation into cell culture because freezing and thawing reduce the ability to recover virus. However, if storage time will exceed 5 days, samples should be frozen at -70. Viral culture procedure takes 3 to 10 days, and classically, embryonated eggs and primary monkey kidney cells are the methods of choice for influenza viruses, but other cell lines can be used. Inoculated cell cultures are incubated in the absence of serum, which may contain non-specific viral inhibitory factors, and in the presence of trypsin, which cleaves and activates the HA so that replicating virus will spread throughout the culture. Cell cultures can be tested for the presence of virus by hemadsorption 3 to 5 days after inoculation, or the culture fluid can be examined for virus after 5 to 7 days by hemagglutination or immunofluorescence. If the results are negative, a passage is made into fresh cultures. This passage may be necessary because primary viral isolates are often fastidious and grow slowly. Viral isolates can be identified by hemagglutination inhibition, a procedure that permits rapid determination of the influenza type and subtype. To do this, reference sera to currently prevalent strains must be used. Hemagglutination by the isolate will be inhibited by specific anti-serum to the homologous subtype. For rapid diagnosis, cell cultures on cover slips and shell vials may be inoculated and stained 1 to 4 days later with monoclonal antibodies to respiratory agents. Rapid viral cultures can also be tested by RT-PCR to identify a cultured agent. It is also possible to identify viral antigen directly in exfoliated cells in nasal aspirates using fluorescent antibodies. This test is rapid but is not as sensitive as PCR or viral isolation. It does not provide full details about the viral strain and does not yield an isolate that can be characterized. Serology, antibodies to several viral proteins are produced during infection with influenza virus. And this immune response against the HA glycoprotein is associated with resistance to infection. Routine serodiagnostic tests in use are based on the HI and enzyme-linked immunoabsorbent assay, or ELISA. Paired acute and convalescent sera are necessary because normal individuals usually have influenza antibodies. A fourfold or greater increase in titer must occur to indicate influenza infection. Human sera often contain non-specific mucoprotein inhibitors that must be destroyed before testing by HI. The HI test reveals the strain of virus responsible for infection only if the correct antigen is available for use. Neutralization tests are the most specific and the best predictor of susceptibility to infection, but are more unwieldy, and more time-consuming to perform than the other tests. ELISA is more sensitive than other assays. Complications may be encountered in attempting to identify the strain of infecting influenza virus by the patient's antibody response because anamnestic responses frequently occur. For epidemiology, influenza viruses occur worldwide, which causes annual outbreaks of variable intensity. And it is estimated that annual epidemics of seasonal influenza cause 3 to 5 million causes of severe illness and 250,000 to 500,000 deaths worldwide. Economic costs have been estimated at $10 to $60 million per million population in industrialized countries, depending on the size of the epidemic. The three types of influenza vary markedly in the epidemiologic patterns. So type C is the least significant because it only causes mild sporadic respiratory disease, but not epidemic influenza. Influenza B sometimes cause epidemic, but it's the influenza type A which can sweep across continents and around the world in massive epidemics called pandemics. The incidence of influenza peaks during the winter. So a continuous person-to-person chain of transmission must exist for the maintenance of the agent between epidemics. And some viral activity can be detected in large population centers throughout each year, indicating that the virus remains endemic in the population and causes a few subclinical or minor infection. For antigenic change, periodic outbreaks appear because of antigenic changes in one or both surface glycoproteins of the virus. When the number of susceptible person in a population reaches a sufficient level, the new strain of virus will cause an epidemic. The change may be gradual, that's why it's known as antigenic drift, because of point mutation reflected in alterations at major antigenic site on the glycoprotein, or drastic and abrupt, which is antigenic shift, owing to the genetic reassortment during the co-infection with an unrelated strain. So all three types of influenza virus exhibit antigenic drift. For the antigenic shift, it is mostly the influenza type A lang, because type B and C are restricted to humans, unlike the influenza A virus that is not just found in humans but also in animal and bird populations. So we can be infected with those viruses, influenza type A viruses that comes from other animals, and this will cause a genetic reassortment from the original influenza virus type A that was also infected in the individual. Periodic outbreaks appear because of, influenza outbreaks occur in waves, although there is no regular periodicity. Wait, okay. Yeah. Periodicity in the occurrence of epidemics. The experience in any given year will reflect the interplay between extent of antigenic drift of the predominant virus and waning immunity in the population. The period between the epidemic waves of influenza A tends to be in 2 to 3 years. The inter-epidemic period for type B will be longer, which is 3 to 6 years. Every 10 to 40 years, when a new subtype of influenza A appears, a pandemic results. So the H1N1 subtype re-emerged in 1977 because it first emerged in 1918. But in 1977, there was no epidemic that happened. In early 2009, there's this novel swine origin H1N1 virus that appeared and reached pandemic spread by mid-year. It was a quadruple reassortant, which contains genes from both North American and Eurasian swine viruses, as well as from avian and human influenza viruses. The virus was readily transmissible among humans and spread globally, causing more than 18,000 deaths. The severity of illness was comparable to that of the seasonal flu. The pandemic virus, named H1N1 pandemic 09, has become a seasonal influenza virus which continues to circulate with other seasonal viruses and appears to have displaced the previously circulating H1N1 strain virus. Surveillance for influenza outbreaks is necessary to identify the early appearance of new strains, with the aim of preparing vaccines against them before an epidemic occurs. That surveillance extends into the animal populations like the birds, pigs, and horses, since they also have the influenza virus type A that can also infect humans. Isolation of a virus with an altered HA in the late spring during a mini epidemic signals a possible epidemic the following winter. This warning sign, termed a herald wave, has been observed to precede influenza A and B epidemics. Avian influenza. Sequence analysis of influenza A viruses isolated from many hosts in different regions of the world support the theory that all mammalian influenza viruses derived from the avian influenza reservoir. So of the 18 HA subtypes that is found in birds, only a few have been transferred to mammals, and for humans there are 6: H1, H2, H3, H5, H7, and H9. The same pattern also holds for the NA. There are 11 known subtypes of NA for the birds, but there's only 2 that are found in humans, which is N1 and N2. So avian influenza ranges from inapparent infections to highly lethal infections in chickens and turkey. So most influenza infections in ducks are virulent. So in other words, they don't cause diseases to ducks. Influenza viruses of ducks multiply in cells lining the intestinal tract and are shed in high concentrations in fecal material into water, where they remain viable for days or weeks, especially at low temperatures. It is likely that avian influenza is a waterborne infection that moving from wild to domestic birds and pig. There is also evidence that supports the model that pigs serve as a mixing vessel for the reassortants because their cells contain receptors that are being recognized by both human and avian viruses. So yun, the pigs can act as an intermediate host. So this it's in the pigs where genetic reassortment often occurs. The pandemic strain of 2009 was a novel reassortant that contained swine origin viral genes as well as those from avian and human influenza viruses. School-age children are the predominant vectors of influenza transmission. Crowding in schools favors the aerosol transmission of the virus, which children take the virus home to their families. In 1997, in Hong Kong, first documented infection of humans by the AH5N1 occurred. The source was the domestic poultry, and by 2006, the geographic presence of the H5N1 avian influenza virus in both wild and domestic birds had expanded to include many countries in Asia, Africa, Europe, and in the Middle East. So these outbreaks were the largest and most severe on record. So of about 425 laboratory confirmed human cases by May 2009, more than half were fatal. So far, isolates from the human cases have contained all RNA gene segments from the avian viruses, which indicates that the avian viruses had jumped directly from bird to human. So there's no, as mentioned in the previous slides, the model that states that the swine is the reservoir host was bypassed. All evidence to date indicates that close contact with deceased birds has been the source of human H5N1 infection. The concern is that given enough opportunities, the highly pathogenic H5N1 avian influenza virus will acquire the ability to spread efficiently and be sustained among humans either by reassortment or by adaptive mutation. And this would result in a devastating influenza pandemic. So PCR technology has yielded gene fragments of influenza virus from archival lung tissue specimens from victims of the 1918 Spanish flu epidemic. The complete coding sequences of all 8 viral RNA segments have been determined, and the sequences document that it was an H1 influenza A virus. It appears that the 1918 virus was not a reassortant, but was derived entirely from an avian source which adapted to humans. Using reverse genetics, an infectious virus containing all the gene segments from the 1918 pandemic virus was constructed. In contrast to ordinary influenza viruses, the 1918 virus was highly pathogenic, including being able to kill mice rapidly. The 1918 HA and polymerase genes appear to be responsible for the high virulence. Now for the prevention and treatment by drugs, amantadine hydrochloride and an analog rimantadine, classed as adamantane drugs, are M2 ion channel inhibitors for systemic use in the treatment and prophylaxis of influenza A. There's also the NA inhibitors, zanamivir and oseltamivir, approved in 1999, and peramivir in 2014, which are useful for treatment of both influenza A and influenza B. To be maximally effective, these drugs must be administered very early in the disease. Resistant viruses emerge more frequently during the use of the M2 inhibitors rather than NA inhibitors, and more frequently in children than adults. Resistance to oseltamivir has been associated with H275Y mutation in NA gene. During the 2013 to 2014, all circulating influenza viruses were resistant to M2 inhibitors, but most were sensitive to NA inhibitors. Depending on the susceptibility of the predominantly circulating strains, subtyping may be useful to determine optimal therapy. Inactivated viral vaccines are the primary means of prevention of influenza in the United States. However, certain characteristics of influenza viruses makes prevention and control of the disease by immunization especially difficult. Existing vaccines are continually being rendered obsolete since the viruses always undergo antigenic drift and shift. And surveillance programs by government agency and the World Health Organization constantly monitor subtypes of influenza circulating around the world to promptly detect the appearance and spread of the new strains. A major advancement for the prevention and control against the influenza virus would be the ability, if we are able to design a vaccine which could stimulate an antibody response that is broadly effective against many subtypes of influenza. Although protection can reach from 70% to 100% in healthy adults, frequency of protection is lower among the elderly and among young children. These are other problems that can be considered in the prevention and control. Since even though if we will give vaccine, it will still be lower among the elderly and young children. And inactivated viral vaccines usually do not generate good local IgA or cell-mediated immune responses, and the immune response is influenced by whether the person is primed by having had prior antigenic experience with an influenza A virus of the same subtype. So for the preparation of inactivated viral vaccines, inactivated influenza A and B virus vaccines are licensed for parenteral use in humans. Federal bodies and the WHO makes recommendations each year about which strain should be included in the vaccine. The vaccine is usually a cocktail containing 1 or 2 type A viruses and 1 type V virus of the strain isolated in the previous winter's outbreak. That's why flu vaccines, we need to have flu vaccines every year, since we are not sure whether that type of virus, of influenza virus, was the one that was administered to us in the previous year. Selected seed strains are grown in embryonated eggs, the substrate used for vaccine production. Sometimes the natural isolates grow too poorly in eggs to permit vaccine production, in which case a reassorted virus is made in the laboratory. The reassorted virus, which will carry the gene for the surface antigen of the desired vaccine with the replication genes from an egg-adapted laboratory virus, is then used for vaccine production. A cell-based vaccine using animal cell culture became available in 2012, which overcomes some limitations of egg-based production. Virus is harvested, purified, concentrated by zonal centrifugation, and inactivated with formalin or beta-propiolactone. The quantity of HA is standardized in each vaccine dose, which is more than 15 micrograms of antigen. But the quantity of NA is not standardized because it is more labile under purification and storage condition. Each dose of vaccine contains the equivalent of about 10 billion virus particles, and vaccines can be either whole virus, subvirion, or surface antigen preparation. When we say whole virus, of course, it's inactivated virus. For subvirion, It contains purified virus disrupted with detergents, and the surface antigen vaccines contains purified HA and NA glycoproteins. For live virus vaccines, so first, of course, we need that the virus must be attenuated first, so as not to induce a disease. It is designated to prevent. Okay. In view of the constantly changing face of influenza viruses in nature and the extensive laboratory efforts required to attenuate a virulent virus, the only feasible strategy is to devise a way to transfer defined attenuating genes from an attenuated master donor virus to each new epidemic or pandemic isolate. A cold-adapted donor virus that is able to grow at 25 degrees Celsius but not at 37, the temperature of the lower respiratory tract, should replicate in the nasopharynx, which is a cooler temperature. A live attenuated, cold-adapted, temperature-sensitive, trivalent influenza virus vaccine administered by nasal spray was licensed in the US in 2003. It was the first live virus influenza vaccine that was approved in the United States, and it is also the first nasally administered vaccine. Annual influenza vaccination is recommended for all children ages 6 months to 18 years and for high-risk groups. So these include individuals at increased risk of complications associated with influenza infection, so like those with chronic heart disease, those with asthma or metabolic or renal disorders, those in the home for the aged, persons infected with HIV, and those aged 65 years and older. And also, of course, those persons who take care of the high-risk groups. The only contraindication to vaccination is of course the allergy to egg protein, since some of the flu vaccines are derived from egg proteins. Although the cell-based vaccine, as I mentioned in the previous slide, overcomes this limitation. So if you are allergic to eggs or chicken, then make sure that the flu vaccine that you are going to administer to yourself would be cell-based. So when the vaccine strains kasi are grown in eggs, some egg protein antigens are present in the vaccine, which causes the allergy. The live virus intranasal vaccine is not currently recommended for individuals in the high-risk groups because live virus, so it may cause, if the individual is immunocompromised, then it can cause symptoms. Prevention by hand hygiene. Although transmission of influenza virus occurs primarily by aerosol spread, hand transmission also is potentially important. And there are studies that shows that hand washing with soap and water or the use of alcohol-based hand rubs is highly effective at reducing the amount of virus on human hands. So that ends for this topic, the orthomyxovirus. We're going to talk about the paramyxovirus